friends, this is part 2 of multiple choice question discussion of biotechnology. The first question is most important method currently utilized for haploid production include the options are anther or pollen culture, ovule culture, bulbosum technique. All these techniques are utilized for haploid production, so the option is D, all the above. Bulbosum technique is options are chromosome elimination, chromosome eliminating following interspecific hybridization, chromosome elimination following intraspecific hybridization, and none of the above. Here the answer is chromosome elimination following interspecific hybridization, because in bulbosum technique the cultivated barley chromosome complement will be remained and bulbosum portion of chromosomes are eliminated, and hybridization is also involved here between cultivated barley and hordeum bulbosum, that is hordeum vulgare into bulbosum. Bulbosum technique is used for haploid production. The development of numerous pollen plantlets in anther culture of Datura inoxia was first reported by Guha and Maheshwari. The process of parthenogenesis is embryo development from unfertilized egg. Parthenogenesis meaning is the embryo formation from embryo sac. There is one more term known as parthenocarpy. Don't get confused between parthenogenesis and parthenocarpy. Parthenocarpy is the seedlessness condition. Capacity for haploid production declines with age of donor plant. That means when we cross the hordeum vulgare into bulbosum, bulbosum is the donor. That means one complement is eliminated here. The as the age begins, that means increasing the age leads to declining the production of number of haploids. In pollen culture, isolation of pollen grains from the cultured anthers is by mechanical method and float culture method. Pollen dimorphism is exhibited by tobacco, wheat, barley, and all. In vitro endogenesis is promoted by pre-treatment of cultured anthers or pollen grains like temperature shock, centrifugation, gamma irradiation. Addition of dash found to increase the success rate of androgenesis. Androgenesis means the formation of embryo by the with the help of male reproductive part. Here, this is increased by ethyl, sucrose, or nutrients specific to certain genotypes. Ovule culture is practiced in the members of families like Malvaceae, Fabaceae, Cruciferae. Ovule culture is mainly tried in in cases like where embryo aborts very early or embryo culture is not possible due to difficulty of the excision at a very early stage. Ovule culture is first attempted in Orchidaceae. Remember, ovule culture was first attempted in Orchidaceae. Ovule culture was first attempted by Nimato and Sagawa. Embryo culture is used. The options are to overcome embryo abortion, to overcome seed dormancy, and embryo rescue in distant hybridization. All these are used in embryo culture. Ovary culture is first reported by San Neom. San Neom, he is the father of gynogenesis. Okay, ovary culture is first reported in barley. Embryo culture is first reported in Raphanus, and this was reported by Hanning. Two things are very important here. Ovary culture was reported by San Neom in barley, and embryo culture by Hanning in Raphanus. The success of embryo culture depends on developmental stage of the embryo at isolation, growth condition of the mother plant, then composition of the nutrient medium. Growth hormone not used in embryo culture medium is auxin. Apart from that, all the other Hormones like gibberellin cytokinins are used in embryo culture. Embryo implantation technique was proposed by Pissarro and Vinogradova. Remember, embryo implantation technique was proposed by Pisava and Vinogradova. The embryo culture is used for embryo rescue, monoploid production, then to overcome the seed dormancy. Bulbosome technique is used for embryo rescue. In vitro fertilization can take place through stigma fertilization, placental fertilization, then fertilization of an isolated ovule without a placenta. Stigma fertilization is successful in Nicotiana rustica, Zea mays, and Pisum sativum. Placental fertilization is successful in Caryophyllaceae, Zea mays, then Gossypium. Conditions necessary for in vitro fertilization include physiological state of the pollen and ovule, morphological state of pollen and ovule, then nutrient medium. Next is the endosperm is dash in nature. It is always triploid. Even alluron layer is also in the same condition. That means the triploid state. Endosperm is product of double fertilization. Triploid nature of endosperm is the characteristic feature of angiosperms. Important characteristics of angiosperm tissue are chromosomal variations, polyploidy, chromosomal bridge, and leg guards. Remember, characters of endosperm tissues are chromosomal variation, polyploidy, chromosome bridges, and leg guards. Successful organogenesis was achieved from endosperm callus tissues of Racinus communis, Oryza sativa, then Pyrus malus. Endosperm culture is successful in Euphorbiaceae, Santalacea, and Laurentheaceae. Euphorbiaceae is your rubber or castor family, then Santalacea is sandalwood family, and Laurentheaceae is Laurenthus family. Triploidy is exploited in apple, banana, watermelon. It is most popular in apple as well as watermelon and bananas. All the currently cultivated bananas are triploid because they are seedless. Vegetative propagation of plants is labor intensive, low in productivity, and seasonal. Detailed account of stages of micropropagation was given by Fassard. Remember, detailed account of stages of micropropagation was given by Fassard. Here I am not reading the other choices because it may cause confusion in you. Detailed account of micropropagation stages was discussed by Fassard. The benefits of micropropagations are rapid multiplication of superior clones, multiplication of disease-free plants, then cost-effective process. Somaclonal variation arises as a result of chromosome structural changes like deletions and duplications, gene mutations, then transposons. Methods involved in the in vitro conservation of germplasm are cryopreservation, cold storage, then low pressure and low oxygen storage. Cryopreservation include the usage of solid carbon dioxide at minus 79 degrees Celsius, vapor nitrogen which is utilized at minus 150 degrees Celsius, and liquid nitrogen which is utilized at minus 196 degrees Celsius. And the most popular method of cryopreservation is liquid nitrogen at minus 196 degrees Celsius. Major benefits of synthetic seeds are easy to store without viability loss, easy to handle, and can be directly sown in soil. Tissue culture is used for vegetative multiplication to get the viral free plants. Then tissue culture has potential application in reduction of production of somatic embryos, organelle and cytoplasm transfer, then genetic transformation. Virus-free plants in tissue culture are produced by meristem tip culture. Remember, virus-free plants in tissue culture are produced by meristematic stem culture. That means the meristematic stems are young growing shoot buds that are free of viruses. Tissue culture is used for production of viral-free plants, rejuvenation of old plant materials, and hybrid production. Cryopreservation of tissue culture material in liquid nitrogen is at, as I told you, it is minus 196 degrees Celsius. Dimethyl sulfoxide is used as cryoprotectant in tissue culture. Term somaclone was proposed by Larkin and Scrocraft. Somaclonal variation include aneuploids, sterile plants, and morphological variants. The usefulness of somaclonal variation was first demonstrated through the recovery of disease-resistant plants in potato and sugarcane. Epigenetic variation include non-heritable phenotypic variation, temporary variation, and ultimately it is reversible. Epigenetics means change at the surface level, not at the DNA base level. That may not lead any basic changes. So ultimately it is reversible, temporary, and non-heritable phenotypic variation is the cause of this. Ploidy changes occur due to endomitosis, endoreduplication, and spindle fusion. Remember, ploidy changes occur due to endomitosis, endoreduplication, and spindle fusion. Gross structural rearrangements like dash are major causes of somaclonal variation. The variations are deletion duplications, inversions, and translocation. Ono is a somaclonal variety of sugarcane. Scarlet is a somaclonal variety of sweet potato. Velvet rose is somaclonal variety of geranium. Then Sigma is somaclonal variety of alfalfa. Bio 13 is somaclonal variety of Citronella java. Varuna is somaclonal variety of Brassica juncea. Now, the limitations of somaclonal variation are applicability only to those species which regenerate complete plants out of cell cultures, reduced or no regeneration potential, undesirable features like reduced fertility, growth, and even overall performance. Gels used for synthetic seed production include carrageenan and gelrite, sodium pectate, and gorgum. Incorporation of activated charcoal in the synthetic seed coating improves conserving the vigor of the encapsulated somatic embryos, increasing respiration of somatic embryos, then retaining nitrogen within the hydrogel capsule and slowly releases them to the growing embryo. These all are the activities of activated charcoal in synthetic seed coating. The artificial seeds can be used for specific purposes like multiplication of non seed production plants, then propagation of male and female sterile plants for hybrid seed production, then propagation of polyploid plants with allied traits. Plant derived from a somatic embryo is referred as seedling and embling. Somatic embryos are bipolar structures, have apical and basal meristematic regions, then capable of forming shoots and roots. Synthetic seeds are of hydrated or desiccated type. Hydrated synthetic seeds are developed by Redenberg. Desiccated synthetic seeds are developed by Kim and Janik. These two things you need to remember. Hydrated synthetic seeds are developed by Redenberg. Then desiccated synthetic seed was proposed by Kim and Janik. Hydrated artificial seeds are sticky, dry rapidly in the open air, then difficult to handle on a large scale. Gels used for synthetic seed production include agar alginate, polycode 2133, then carboxymethyl cellulose. Gels used for synthetic seed production include carrageenan, then gelrite, sodium pectate, and gorgum. Alginate hydrogel is frequently selected as a matrix for synthetic seed because of its moderate viscosity and low spinability of solution, low toxicity for somatic embryo and quick gelation, then low cost and biocompatibility characteristic. Gels used for synthetic seed production include carrageenan and gellrite, sodium pectate, and gorgum. Incorporation of activated charcoal lead to conversion and vigor of the encapsulated somatic embryo, increasing respiration, then retaining nutrients within the hydrogel capsule and slowly releases them to the growing embryo. Artificial seed can be used for specific purposes like multiplication of non seed producing plants, propagation of male or female sterile plants for hybrid seed production, then propagation of polyploid plants with allied traits. The variability in chromosome number in somatic hybrids could be due to multiple fusion give a higher chromosome number, asymmetric hybrids, than somaclonal variation in cultured cells. Somatic hybridization is used in genetic recombination in asexual or sterile plants, overcoming barriers in sexual incompatibility. The first somatic hybrids are produced in Datura inoxia, and this was produced by Cedar. The first to resynthesize rapeseed in vitro using protoplast fusion was Senk. Here again you need to remember two scientists. The first somatic hybrids are produced by Cedar, then Senk was the first to resynthesize rapeseed in vitro using protoplast fusion. The feasibility of enzymatic degradation of plant cell walls to obtain large quantities of protoplast was demonstrated by E.C. Cocking. Protoplasts were isolated by mechanical method and enzymatic method. The protoplast isolation by enzymatic method was demonstrated by E.C. Cocking. Enzyme used in enzymatic method of protoplast isolation is cellulase and pectinase. Commercial preparations of the enzyme for protoplast isolation were first employed by Takebe and co-workers. I repeat, commercial preparation of the enzymes for protoplast isolation was first employed by Takebe and co-workers. The leaf has been the most favorite source of plant protoplast isolation because isolation of a large number of relatively uniform cells without the necessity of killing the plants. Mesophyll cells is loosely arranged and the enzymes have an easy access to the cell wall. Most widely used osmotica for protoplast isolation was sorbitol and mannitol. Culturing of protoplasts in liquid media include drop culture, microchamber culture, then multiple drop array technique and microdroplet culture. Culturing of single protoplast in liquid media is done by microdroplet culture. Minimum planting density, that is commonly called as MPD, in protoplast culture is maintained by feeder layer, nurse culture and reservoir media. First report of plant regeneration from isolated protoplasts was from Nicotiana tabacum. First report of plant regeneration from isolated protoplast was done by Takebe for protoplast fusion NaNO3, then that is sodium nitrate, high pH, high carbon calcium concentration, then polygalactouride, that is PEG concentration is important. The mechanism of chemical fusion of protoplasts consists of agglutination, membrane fusion, and rounding off of fused protoplast. Electrofusion of protoplast is rapid and simple, synchronous, more easily controlled. rDNA technology has provided the means to achieve amplification of cloned genes, fractionation of individual DNA components of complex genome, then potential to create new genetic combinations. Other terms that can be used to describe rDNA technology are gene manipulation, gene cloning, and genetic modification. Essential steps in rDNA technology are for cDNA synthesis. The eukaryotic mRNA is used as a template to generate DNA. Enzyme reverse transcriptase is also called as RNA dependent RNA polymerase. The process of reverse transcription is also called as taminism. Chemical synthesis of DNA is by phosphodiester and phosphotriester method. The first method used to artificially synthesize the DNA was phosphodiester, but phosphotriester method is most popular. The tool for cutting DNA molecules is restriction endonuclease, that is an enzyme. Tool for joining DNA molecules is ligases, that is also known as molecular glue. Important tools used in rDNA technology include enzymes for DNA manipulation, vectors, expression hosts. rDNA technology is used for gene cloning, DNA sequencing, creation of transgenic plants and animals. The restriction enzymes are grouped into three types, so type 1, type 2, type 3. The restriction enzyme used abundantly in rDNA technology is type 2 because that is sequence specific. Restriction enzyme with star activity are E. coli, BAM-HI, then SAL1. Enzymes used in rDNA technology are nucleases, restriction enzymes, DNA ligase. Enzymes used in rDNA technology, other enzymes are kinase, phosphatase, reverse transcriptase, and other enzymes like terminal deoxynucleotide transferase, the nucleotide transferase, then RNSP and reverse transcriptase. Group of enzymes used in rDNA technology which cleaves or cut the genetic material are nucleases. Nucleases act both on DNA and RNA. The nomenclature of restriction enzymes was proposed by Smith and Nathans. Remember, nomenclature of restriction enzyme was proposed by Smith and Nathans. Restriction enzymes are grouped into three types. The restriction enzyme with star activity are E. coli, BAM-H1 and SOL1. DNA ligase joins the DNA molecule covalently by catalyzing the formation of phosphodiester bonds between adjacent nucleotides. DNA ligase is isolated from E. coli and T4 bacteriophage. DNA ligase isolated from and is widely used in E. coli and T4 bacteriophage. Reverse transcriptase isolated from and is widely used in E. coli and T4 bacteriophage. RNSP specifically cleaves at the 5' end as well as 3' end of RNA. Vectors used in rDNA technology should possess the characters like autonomous replication, small size, possess one or more genetic markers to identify their easy movement in the next generation. Plasmid DNA is self-replicating, double-stranded, and circular. Plasmids are grouped into two major groups. Conjugative plasmids have only transfer genes, and they have both. Conjugative plasmids have tra genes as well as mob genes, that is transfer gene and mobilizing gene. Non-conjugative plasmids have only mobilizing or mob genes. Relaxed plasmids are also called as high copy number plasmids. Stringent plasmids are also called as low copy number plasmids. Remember, relaxed plasmids are also called as high copy number plasmids and stringent plasmids are also called as low copy number plasmids. Plasmids that carry specific sets of genes for the utilization of unusual metabolites are called as degradative plasmids. I repeat, plasmids that carry specific sets of genes for the utilization of unusual metabolites is called as degradative plasmids. Plasmids without any apparent function coding genes are called as cryptic plasmids. That means they doesn't code for specific function are called as cryptic plasmids. Plasmids capable of promoting their own transfer to a wide range of hosts are called as promiscuous plasmids. I repeat, plasmids capable of promoting their own transfer to a new wide range of hosts means they have capacity themselves to promote to other wide range of hosts is promiscuous plasmids. PBR322 plasmid was created by Bolivar Rodriguez. Resistant genes in PBR322 plasmid is ampicillin and tetracycline. Resistant genes in PUC19 plasmid is ampicillin. Phase vector that contains single site for the insertion of foreign DNA are designated as insertional vectors. Remember, phase vector that contains single site for the insertion of foreign DNA are designated as insertional vectors. Phase vectors that contain two cleavage sites and which allow foreign DNA to be substituted for the DNA sequence between those sites are designated as replacement vectors. Plasmids containing phase cos sites are also known as cosmids. Phasmids are also called as phagemids. Plasmids are hybrids formed between plasmids and bacteriophages. Bacterial artificial chromosome or BAC has many of plasmid have the ability to initiate conjugation with adjacent bacteria, have a cloning limit 75 to 300 kb. Yeast artificial chromosomes are artificial chromosomes that replicate in yeast cells, have recognition sites for restriction enzymes, and have ampicillin resistant genes for selective amplification. Yeast artificial chromosomes are the largest of the cloning vectors. Limit is 100 to 1000 kb and have very low efficiency. The larger the plasmid, lower is the efficiency with which it is taken up by the cells, because as the size increases, the movability of the plasmid into the next or the host cell will decrease due to its size. Vectors used in genetic engineering should possess the characters like multiple unique restriction sites, bacterial regions of replication, then marker gene which allow recognition of transformed cells. Agrobacterium TI plasmid is preferred over all the other vectors because wide host range and capacity to transfer genes due to the presence of tDNA border sequences. Natural genetic engineering is Agrobacterium tumefaciens. It causes crown gall. Another species is Agrobacterium rhizogenes, which causes hairy root. Then TI plasmids have four regions in common. I'll just tell you the different regions. A region responsible for tumor induction is TI A region A. Then region B is responsible for replication. Region C is responsible for conjugation and D is for virulence. I repeat, region A is responsible for tumor induction, region B is for replication, C is for conjugation, and D is for virulence. Genes responsible for shooty locus in tDNA region are TMS and TMS2. Genes for rooty locus in tDNA region is TMR. Chemical mediated, that is PEG transfer, is proposed by Kranz. Remember, chemical mediated PEG transfer is proposed by Kranz. Optimum concentration of PEG to the DNA transfer is 15 to 25%. Microinjection method was introduced by Crossway and Rich. Microinjection was introduced by Crossway and Rich. Electroporation method was introduced by Fromm. Particle gun for gene transfer was introduced by Klein. Sonication method was proposed by Xu. Clone is the exact copy of an organism, organ, or single cell. Molecular cloning refers to isolation of a definite DNA sequence or obtaining multiple copies of defined DNA sequence in vivo. Cloning is frequently employed to amplify DNA fragments containing genes, DNA sequences such as promoters, non-coding sequences, then chemically synthesized oligonucleotides. Cloning is mainly used for large-scale protein production or gene therapy. Taq polymerase is thermostable. Restriction enzymes cut DNA at specific locations based on the nucleotide sequences. DNA ligase enzyme unites the DNA segments. DNA ligase creates both covalent bonds between the nucleotide chains. The process of transferring plasmids into new host is called as transformation. Bar genes conferring resistance to bilaphos and phosphonotocin was isolated from streptomycin hygroscopias. Fluorsor is the GM variety of tomato. Fluorsor, the GM variety of tomato, has delayed ripening property. The potential benefits of GM crops include high crop yield, reduced farm cost, and increased profit. Potential hazards are danger of unintentionally introduced allergens and other anti-nutrition factors in foods, likelihood of transgene escaping from cultivated crops to wild relatives, potential for pests to evolve resistance to the toxins produced by GM crops. Disadvantages of blotting techniques include complex, cumbersome, and time consuming. Here, I'll just refresh your knowledge about blotting technique. The technique was introduced by Southern in the year 1975, and there were four methods. One is Southern blotting, then Eastern, Western, and Northern blotting. They have specific applicability. So, disadvantages of blotting techniques are that it is complex, cumbersome, time consuming, no information about regulation and gene interaction, then analysis of one gene or RNA at a time. DNA based molecular markers are widely used in taxonomy, embryology, and genetic engineering. Term DNA fingerprinting was introduced for the first time by Alec Jeffrey. Properties desired and in the year 1985. Properties desirable for ideal DNA markers are highly polymorphic codominance, frequent occurrence in genome. Properties desirable for ideal DNA markers are selective, neutral behavior, easy access, easy and fast assay. Other properties like high reproducibility, easy and fast assay, easy exchange of data between laboratories. Single or low copy probes include RFLP, EST, and RLGS. RFLP markers were used for the first time for the construction of genetic maps by Botstein. RLG markers were first time used for construction of genetic maps by Hatada. Technique in which nucleic acid or proteins are immobilized onto a solid support is called as blotting. Blotting techniques are used to identify unique proteins and nucleic acid sequences. Blotting techniques consist of four separate steps. Southern blot is a method to check for the presence of DNA. Southern blot was invented by Edwin Southern. Northern blot was invented by Alvin, Stark, and Kemp. The method used to check the presence of RNA is Northern blot. Northern blot is used to overexpression of oncogenes and downregulation of tumor suppression genes. The advantage of northern blots are detection of RNA size, quality and quantity of RNA can be measured. Use the probes with partial morphology. Immunoblot is the other name for Western blot because here protein analysis is carried out. Western blot is a method used to check the presence of protein. Then Eastern blot is the extension of Western blot. Northern blot is the extension of Southern blot. Eastern blot is a method used to check for the presence of protein post-translational modification. Remember, Western blot is for the detection of presence of specific protein, whereas Eastern blot is for protein post-translational modification. Eastern blot was invented by Neil Burnett. Friends, the information furnished by me I hope is informative to you. If you like this video, please do not forget to keep on commenting, subscribing, liking, and sharing. Thanks for watching.